Okay, you have given me multiple sharing?
Yes, sir.
Okay. Just tell me whether it is, we are able to see the screen?
Just a minute, sir. It's yeah, yes, sir.
I have some problem because when I put PPT, it doesn't go to the next slide. So what I will do, I have already converted into PDF, so I'll be operating with the PDF.
Okay, sir.
Okay.
Sir, just a minute. I request Amruta madam to.
Yeah, I mean, I'm
Yeah.
getting some. Okay, fine. I think. Let me just check it actually whether slide is getting changed.
Sure, sir. Sure.
Okay.
Yeah, it's working, sir.
Yes, yes,
It's working. Fine.
yes, sir.
Yes, sir.
Thank you. Thank you.
Yeah. Shall I start?
Yes, madam.
A very good afternoon to one and all. I am Mrs. Amrita Auraskar, and I am the host for this session. Today, for the second session of this online international faculty development program, we have with us Dr. K.S. Ladda. Dr. K.S. Ladda is a professor in pharmacognosy and phytochemistry, Department of Pharmaceutical Sciences and Technology, ICT Mumbai, since 1989. Sir has 31 years of vast professional work experience and has expertise in extraction and isolation, characterization of phytoconstituents, development of large-scale extraction technologies, standardization of Ayurvedic herbal drugs and formulations, development of Ayurvedic herbal drug formulations, chemical modification of phytoconstituents, scientific studies on Shodhana Prakriya of toxic plants in Ayurveda. Sir has over 200 publications in national and international journals and 7 books to his credit. Sir has total 16 patents with 1 US patent granted. He has published 60 monographs in quality standards of Indian medicinal plants by Indian Council of Medicinal Research, New Delhi. Sir has guided over 75 master students and 25 PhD students till date. He has worked on 24 sponsored projects from government as well as industry. He provides consultancy to many reputed industries like Charak Pharma, Total Herb Solutions, Yukka Enterprises, Tata International, Pidilite Industries, Pitambari, Godrej, to name a few. We are fortunate enough to have such a great, learned, and experienced speaker for today. His experience and contribution to herbal drug industry is huge and commendable. Let us patiently listen to Sir and gather knowledge from his talk on phytochemicals, challenges and opportunities. Over to you, sir.
Thank you, Dr. Amrita. And let me first tender my apology for being late and some technical hitch in joining.
That's absolutely fine, sir. No problem.
I'm sorry, actually, I'm in Baroda at this moment with Bajaj Healthcare. Actually, I have come here actually to undertake project on extraction isolation of opium alkaloid from poppy straw. Okay, so I mean, my apology for a little. I'm not in my hometown.
That's absolutely fine, sir.
Yes. Good morning to everyone. Good morning to everyone. And I'm Radha from Institute of Chemical Technology. And as you know, our name of the institute is Chemical and Technology. So I'll be precisely dealing with the chemicals and the technology, though of course it is of the herbal origin. As regard to the, I mean, topic of the or the theme of the FDP, empowering educators, I think this is very close to my heart actually because I always believed actually that the teacher must be empowered because if the teachers are empowered, the thing gets passed on to the student. And many times, whenever I get an opportunity to interact with the higher up in the institute, I always tell them actually that please give the freedom to the faculty because if you give the freedom, obviously after some time, that sense of the responsibility also comes in. So I always believe actually the freedom should be given to the educator because we work with the mind actually. We are not, I mean, I don't think we are meant for the physical work as such, but we work with the mind. And yes, physical work follows thereafter. So topic is quite good actually, and I'll be dealing with some technological aspect associated with my subject, pharmacognosy. Um, it is also called medicinal natural product. Some terms, which have been used in the past, and now of course we call it herbal technology. So with this, actually, I'll begin with my talk. Yeah, empowering educators, advancement in technology research and entrepreneurship. I think this is all very important theme. Empowering educator, definitely the teacher must be, or the faculty from the Pharma Institute. Of course, since I'm connected with the pharma, I'll be more confined to the pharma faculty, and they should be encouraged. They should be provided with a good facility so that their mind works in the right direction. And afterwards, actually, once you provide this kind of conducive atmosphere to work, definitely, I'm sure everybody want to develop actually, and they will come, they will put their brain in action, do some research, come out with some technology, and if not practice themselves, probably they will help others actually to become entrepreneurs. I will appreciate if some pharmacy people can become, entrepreneurs in the same direction in which they are teaching the subject. That will be the most appropriate thing. But otherwise, also if they are able to encourage others to get into the subject matter, nothing like it. I'm sorry. All right. All right. I will, with this actually, I will deal with the subject matter. Botanicals and herbals, those are the things which I have been talking about it, and I've been dealing about it actually. And world over now, there is a resurgence of herbals in the different forms, whether it is herbals, botanicals, nutraceuticals. So these are the different forms in which it has resurfaced, not just resurfaced, but today it is trending also. Now, if you see entire field, whether it is cosmetic, whether it is a nutraceutical or functional food, I think it is heavily controlled actually by the medicinal plant or nutraceutical plant. Besides, actually, since I'm in pharmacy and majority of us are from the pharma, I think we should also keep our eyes open about the application of whatever we are doing in the other sector. And that is why, in briefly, I have touched upon this slide that yes, plants are also extensively used in the lubricants, perfumes, flavors, natural dyes, spirits, tanning industry, biofertilizer, biopesticide, algal biofuel. So these are the other areas actually where plants are extensively being used, and definitely our focus is more on the medicinal plant, but at the same time when you work actually on certain things, I think you must also keep in mind sometime you may not find appropriate application in the pharma, but the same plant may have the application in the other field. And I think I must tell you, one of my project actually I have undertaken, kala dana. We all have heard about the kala dana, and I remember actually when I was a student, kala dana was taught, and when I started teaching, actually I also started teaching kala dana. There's, yes, it is actually Ipomia hiridacea seed, and it contains resin, and the action is purgative, but it is no longer used. So after the entire information is given, actually we say that it is no longer in use. And I relooked at the same plant actually, and today we have converted this the same kala dana into an important constituent called igdason, which is finding application in other field like sericulture. So just, request to all those in the field, actually they should look beyond uh, medicinal plant, their action. All right, let me just take you through some of the aspect. Morning, I was coming with the manager of the Bajaj Healthcare, and we were just discussing. I said almost 80% of the APIs today, what you see around, those are all either they are directly, came from the plant, or they have been influenced by the plant, the constituents of that. Whether you take, analgesic, or whether you take today's, of course, metformin or the butformin or the fenformin, those are all derived actually from the natural plant. So I will take you in short actually through some of the medicinal application of the plant material. Paclitaxel, all of you know, it's a Taxol, which has been used as the anti-cancer drug. Some more like atropine, digoxin, etoposide, silabine, morphine, those are all still coming from the plant source actually. Carbinoxolone obtained from the glycyrrhizin converted into a succinic acid derivative. Reserpine, hypotensive drug. THC and CBD has been talked about it later on. It's been talked about it actually extensively, but unfortunately in India we are still having a problem. But CBD is now been opened up actually in the US, and I'm sure THC also will be. Both of them are coming from the marijuana or the cannabis. But yes, these are the drugs actually which have been directly derived from the plant. And besides, there are quite a few more. Nutraceutical, as I told you in the beginning, that many plants have been used today actually in the nutraceutical. And some of the sectors where the plants have been extensively used in nutraceutical includes probiotic, protein, amino acids. Today, a lot of protein, plant-derived proteins actually, mainly the legume-derived proteins are trending now. Phytopharmaceuticals and the plant extracts are once again extensively been used in the nutraceutical, whether it is fiber, that is FOS, that is, fructooligosaccharides, specialty carbohydrate, vitamins, prebiotic, carotenoids, omega-3 fatty acids, some of the minerals. So these are all the constituents which has by and large been derived from the plant source and extensively been used in the nutraceutical or the functional food as an ingredients. These are the application-wise actually nutraceutical. The common form in which nutraceuticals are available today in the form of food, feed supplement they call it, beverages, personal care product, animal nutrition, and dietary supplements. So these are the different forms in which they have become very popular. Some of the nutraceutical ingredient, which of course these are some lifestyle problem by which the people are now encountering, and some of the health benefits which are associated with the nutraceutical ingredient, which has been marketed extensively by the companies, include cognitive health, gut health. I mean, our lifestyle has changed actually, less exercise and more sitting job. Heart problems are increasing actually. Bone health, again, lifestyle problem, immunity. Yes, I have played in the mud, but the current generation don't play in the mud actually because the parent does not allow. But now the one study has come actually that yes, more they become and more they play in the older fashion, I think they develop more immunity. Nutrition, weight gain is another problem actually, which slowly become obesity, which wasn't heard in India, now it has become a problem. So these are some of the lifestyle disorder by which nutraceutical have been addressed. Now herbs in the food sector, just to give an idea, annatto is a color actually which has been obtained from Bixa origina, and it has been used actually in giving the color to amul butter or allied butters. Stevia glycoside, which are once again obtained from a plant called Stevia rebaudiana. Today it is extensively been used for the diabetic people. In fact, it has become actually an important ingredient of the sweetmeat targeted for the diabetic people. Lycopene, carotene rather, vanillin is extensively used. Of course, most of the vanillin which has been used in the ice cream industry, majority of it has been coming from the synthetic source. Natural source is pretty expensive. Gallic acid and the gallets are used in the food sector for as a preservative for the preservative. Thumatin, slightly unheard of it actually, but it is African plant from which it has been obtained. Plant is Tomatococcus ganil, and it is a protein sweetener. Obviously, diabetic people are looking for some non-glycemic sweetener, and thumatin do satisfy that need. The only problem associated with the thumatin is that it is a protein, so which it cannot be used with the hot beverages. But with the cold product, definitely it is having a good application. Glycerin, another sweetener which has been permitted by the FDA as well, having a problem. Now, these are some of the phytochemicals which are used in a sector other than food and the pharma. For example, lawson is been used, which is present in the henna leaf actually, is been used for the hair dye, cosmetic industry you can call it. Indigo is been used actually for coloring denim or jeans. Limonin, which is actually a byproduct of citrus oil, because citrus oil comprises of citral, which is an important flavoring ingredient, but almost 80 to 90% consists of the limonin, which is rather unwanted material, and but that is having a good application in as an industrial degreasing agent. And menthol, of course, extensively used in the confectionery. Karanjin, an Indian plant which contains furanochromone derivative, is having insecticidal activity. So is the case with the sesame, which is obtained from the Sesamum indicum or the gingerly seed. Quinine, we have been taught actually cinchona has been included in the syllabus, and we say okay, cinchona contains quinine, and quinine is having the antimalarial action, but currently quinine is no longer used as an antimalarial drug. But quinine extensively is used actually for flavoring. and for imparting bitterness to the liquor. So it has find important application in the liquor industry. Oxalic acid, which has been used in the cosmetic product as a protective. So these are some of the products which have been derived from the plant but used in the other sectors. So this gives an idea about the different sectors where the plants are having application. Now, since the topic is entrepreneurship uh, and the research and the development, I think all good development of the research takes place because of the challenges which people face it. If you look around the success story, most of the people's success story is because of they have faced some hardship, and to overcome that hardship, actually they have found out the solution and they become successful. So it is very important actually that you must see what are the challenges which are existing, because once you know the challenges, then only the solution can be found out. So some of the challenges by which this sector is suffering is, of course, agrotechnology for medicinal plant. Even today, not many medicinal plants are cultivated in India. The story is different in China, where majority of the medicinal plants are cultivated, and that is why you'll find in US market it is more Chinese products which are more popular, because Chinese FDA, they expect actually the drug or the herbs which are used in the formulation must be obtained from the cultivated source rather than from the wild source. Anyway, but in India also now the medicinal plant board, which comes under the Ministry of the Ayush, is now started, encouraging people to get into the medicinal plant cultivation. QC of the raw material and the formulation is still a problem in India, because first of all it is not mandatory. With the result, actually, I don't see many people performing very serious kind of the analysis. Whatever has been performed is very trivial, and with the result, many times we normally complain that the product is of substandard quality or it is not working. And I think to a great extent the answer lies in this particular thing, that we don't have the robust quality control parameters, whether it is raw material or whether it is finished product. And for that you need phytochemicals, which is the area which I'll be touching upon it. Extraction, the cleanup technology, yes, we need actually good amount of extraction technology, because as we know, plants have been used actually for some of the active constituents which are present in them. And increasingly we must make an advancement of how to improve the content of those important constituents which are pharmacologically active, because more you remove actually the unwanted material, you will be able to make it the product more potent, or even for its a potency purpose. Phytochemicals, yes, this is the sector which is now, at least I'm seeing actually it has been now steadily increasing. In the sense, actually, people are increasingly pharmacological people, those who are into the pharmacology, they're increasingly using now, instead of extract, they're using isolated constituents. So is the chemistry people, because there also I have seen many people are now trying to develop derivatives from the natural product, because natural product provides very complicated molecule in the ready-made form. So that provides definitely some advantages. So some development is taking place in the phytochemicals, extraction, and isolation. Isolating new leads is still a big problem actually. I don't think we have been able to come up with a many new lead, because we don't have many people who are having the expertise in reading the spectra of the natural product of the unknown compound. So this is the area where we need to make some more progress actually before we get into it. Pharmacology and the clinical studies, well, it's not my subject, so I won't be able to talk much about that. Development of the modern dosage form, definitely actually, because the newer generation do not like the classical Ayurvedic product or the generic Ayurvedic product. You need to address their requirement in the modern way. And this is what the challenge is actually, that you must come out. There's a Ayurvedic goodness, but in a modern form. So that is the need of the hour. And I think there is a need for the good amount of work need to be undertaken actually in this direction. Old formulation which are there in the Ayurveda. Some of the slides I will simply, flash it and proceed, because, well, there's a scope in every sector. Herbal cultivation, there's a scope, and now I think there's a center actually in the Pune for the Western region, wherein actually it is in the Savitribai Phule University of the Pune University. Dr. Mukad is in charge, and the mandate has been given to them to encourage medicinal plant cultivation in the Western region. And government is also pumping money actually for the cultivation. Unfortunately, not many formerly educated people are entering into this sector. Otherwise, there is a need to enter into this sector, and the need is actually how to improve the important constituent in the medicinal plant, so that with the lesser biomass we'll be able to meet the more need. Herbal raw material, trade and commerce. Again, the same field, actually same problem. Not many qualified people are entering into this field, and with the result, by and large, this field has been managed actually by the traditional people who doesn't have much knowledge. Whatever knowledge they have, actually, it is long experience in the field actually. So I don't say their knowledge is bad, but their knowledge has some limitation. I think those who are having the formal education in the subject, actually, if they enter, definitely they'll be able to make better sense actually in the trade and commerce, because ultimately the quality of herbal product cannot be built at the last stage. It has to start from the initial stage, and this is the first channel actually where people come across the medicinal plant or the product of that. And well, internet, you'll find actually many people are dealing, but those are by and large it has been managed by the traditional people. So there is a need actually that yes, qualified people must enter into that. Research and development is the area where we are all academicians are involved, and though we are working, but I must confess here that we are not working actually in right direction, and we are more haphazard, and by and large actually it is not leading to a very concrete output. So the time has come actually that we must make effort actually to, at the individual level also, and at the joint effort, that we must solve some of the burning problems first, and thereafter get into some newer things actually. Herbal Ayurvedic products, development of the herbal products and the novel drug delivery system in the herbals. This is the need of the hour. And as I told you, the medicinal goodness of the Ayurvedic product must be converted into the modern form so that it appeals to the newer generation. Otherwise, it will remain confined to the old people only. I mean, Ayurvedic products are quite popular with the geriatric rather than other sector. I mean, I'm sorry, I'll be putting a harsh fact, but those are the reality. I think we must accept that. Sooner we accept, it is better. Well, novel drug delivery system, those who have attempted actually, and you can see some product which are existing in the market actually. Right now, there are very few products, but yes, they are there, and there's a need actually. The more products should come into the market in the modern dosage form, then probably I think it will appeal to the newer generation, and probably it will have better circulation. These are some of the advantages of the modern, I mean, a newer delivery system actually enhanced efficacy with a small quantity. It prevents actually the repetitive administration, better patient compliance. As you improve actually the potential of that, definitely it will reduce actually the unwanted material. It has a lesser toxicity because now you're able to manage the dose which has been corresponding to the exact concentration of the actives in that. Bioavailability, in case if there is a problem, it can be managed actually in case if you have a more enriched extract or isolated phytochemicals. All right, I will skip some of these slides actually. Now, one challenge actually is in herbal, what to analyze and how to analyze is the major problem. Well, as per syllabus is concerned, actually we know, at least in most of the herbals, what we are talking about or what we are teaching, we know the major constituent which has come out. Like in case of Rolfia, we know reserpine is a major constituent, both biologically active also, as well as concentration wise also. But besides reserpine, Rolfia also contains 35 more alkaloids. Unfortunately, not much work has been done on that. The reason actually because we don't have those constituents isolated in the sufficient quantity at affordable price. With the result, actually, the activity in other direction has not progressed much. So the time has come actually that you must be able to make it available. Then only we'll be able to find out the newer application of those other constituents which are present in, like Rolfia, many other plants have the many other constituents which need to be worked upon. Anyway, the major problem with the industry facing actually, especially the either whether you have a monoherbal product or polyherbal product. In polyherbal, it becomes all the more complex. What to analyze and how to analyze, and this problem becomes very severe actually when you take classical Ayurvedic product which comprises of let's say 20 ingredient, 30 ingredient. Then the question arises: what to analyze and how to analyze. What to analyze, because we have to be practical that we cannot go on analyzing each and every ingredient which has been put into the formulation. If you take any classical cup syrup of the Ayurveda, it contains not less than 10 ingredient of the herbal origin. And if you have to analyze all these 10 ingredients, it becomes very difficult. Plus, it is taxing also in terms of the cost. So, how to address this problem without much difficulty? So, this I will explain you with one slide. Actually, this is a cup syrup. what I did actually, I just compiled the data of the 4 cup syrup which are existing in the market, and I simply compiled the data as per their label claims. And when I did some work on that, what is something which everybody has used it, and if you see here, only 4 ingredients which are there actually, which are common with all the marketed formulations. And then I had drawn my own inference that yes, in case if you want to make cup syrup, I think these 4 ingredients are must ingredient. So what to analyze? I think the question gets addressed here by saying that yes, you can priority wise. These are the 4 drugs which need to be kept on the priority for the analysis purpose. So when I look at it, actually the cup syrup, I feel actually there the 4 ingredient, which is Kantakari, Sodium xanthocarpum, or licorice, which is Glycida glabra, or Tulsi, Osmium sanctum, and Vasaka, that is Agathora vesica. These are the 4 ingredients, and if you look at their pharmacological action wise also, they appear to be appearing that they are associated with the different pharmacological action. And with the result actually to undertake what to analyze, the answer is these 4 constituents must be analyzed actually in the cup syrup. And how to analyze? Now the next question arises, how to analyze? If you see most of the current practices of whether raw material or the finished product, they normally analyze by very trivial methods like ash value, extractive value. Those are the values which are normally been used, parameters are normally been used actually for analysis. But according to me, those are not the right parameters, because practically every drug, whatever, whether it is of the same origin or the different origin, will be able to comply to that requirement. So the right analysis will be, finding it out the exact constituent which is responsible for the biological action and the concentration of that. I'm searching actually, even today I'm searching for the vaccine which has been responsible for the anti-tissue action, but unfortunately I don't think we have been able to fix it up the right dose of the vaccine to be given in the 5 ml of the cup syrup, which will be effective. So this is an opportunity that pharmacologists must fix up the dose of most of this constituent, at what dose this will be working as an anti-tissue or the expectorant formulation. So my request actually that what to analyze, of course we have decided. How to analyze, we must use the modern method, because those methods are more precise, more accurate. You need very less constituent actually, but they are reproducible, and with the result we must employ modern analytical methods, whether it is for the qualitative analysis or the quantitative analysis. Now gone are the days in the 1955 pharmacopoeia where every assay method was either gravimetry or the titration. In 1985 pharmacopoeia, it was all colorimetry or the spectrophotometry. 2005 onwards, we have all the methods actually or the HPLC, and I think that is the answer, that we must evaluate the phytochemical constituent which are present in the herbs by the HPLC and determine the exact concentration of important or the actives in that. This is one of the exercise what we did actually, polyherb formulation, and the challenge was thrown to us that this is a 9 herb product, and company wanted to, because they used the extract and they wanted to show that yes, all the 9 herbs are represented in the extract and they are represented as per the label claim. Because Radix astragalus, this is a Chinese product, but it was marketed actually from Singapore, and the mandate was given to me. So what we did actually, we took out one marker compound which was specific for that particular plant material. The things like Bristedia, astragaloside, conchinone, cuniflorin, ferulic acid, hydroxysaffar yellow, amygdalin, that is cyanogenic glycoside, salvinoleic acid, tenifolium, and beta-azarone. So these are the constituent which is representing these herbs which have been mentioned above. And what we did initially, we showed the presence actually by HPTLC. So you can see here the astragaloside presence is being shown. Here the 8 formulation which were found to be biologically active. The company was making this product actually, and after making this extract, they used to subject to the biological model, and they have one biological model. Unless it passes through, they were not releasing the batch. And invariably, what they were finding it actually, that out of 3 batches, 1 to 2 batches usually gets failed. So they were worried about that. So they asked me actually what to do. So I suggested them, why don't we fix it up actually the constituent which are present in these herbs. And for that, I asked them actually, you give me all the batches which has been passed, so that we can create a window of constituents which are present in them. And thereafter, I think once we have a window, then probably once we make the next batch, and if we get in the same window, there are chances that that batch will be able to pass to the biological model as well. And that is what precisely we did. So we showed the presence by first by HPTLC. So qualitative presence has been shown, and thereafter we also showed the presence, quantitative presence actually by way of HPLC. And then we were able to fix it up actually the window for every constituent which are present, which were representing the each drug, and it has helped them actually to minimize the rejection actually of the batches. This is another study what we have done, and we have been granted actually US patent on that. Now, what we suggested that we will be using, it was the four herbs, a product which was comprising actually of hydroxy safflower yellow, accurate atarunavi that contains beta-sarone, and amygdalin, which was from the bitter almond, or they have a semen persica. And out of which, if you see the constituent, actually hydroxy safflower contains water-soluble constituents, Carthamus tinctorius that is, and amygdalin contains glycoside, which has got solubility both in water as well as non-porous solvent, whereas beta-acerone, which is from the accurate atarunavi, was actually it is actually a volatile oil, and which is having more solubility in a non-porous solvent rather than the porous solvent. So when we recommended actually that the concentration of the extraction media must be 70% alcohol and 30% water, so they are the reason. And then we showed in terms of the HPTLC that yes, when you extract the first track is of the standard, second track actually is of extracting the ethanol, third track is at the ethanol, the water mixture, and the fourth track is only the water. So if you see the first slide, where you'll find the middle track which comprises of both water and the alcohol mixture, there you'll find the spots which are quite intense. In this case, this is actually a amygdalin, this is safflower yellow. So here you see actually, since the content is water soluble, it is not found to be present actually in the ethanol, but it is showing presence only in the water and the ethanol mixture as well as only water. In this case, beta-acerone, the presence is shown only in ethanol and ethanol in water mixture, but not in water. So on the basis of these three, we were able to convince them that yes, this is the combination of the water and the alcohol, which gives all the ingredients which are present in all these three herbs actually. Any one water or either water or the alcohol does not make much sense because they do not extract actually all the relevant constituents from that. So this is how one can prove actually that why the importance of the combination of the solvent. All right, so that was about extraction related study or rather actually the analysis related thing. Now, extraction of herbals and the isolation of the phytochemicals, this is the area, and according to me, this provides a good opportunity to become the entrepreneurship, because if you look at most of the phytochemicals, probably I think I have one slide on that. Yeah, these are the phytochemicals which are known by the different terminal or the different names. I'll call it, in pharmacognosy we call it synonyms. So secondary metabolite or the plant analyte or the phytoconstituents or bioactive compounds, analytes. So they are the various terms which have been used, but they all are synonymous. Well, let me just give you some idea about these phytochemicals, derivatives and the synthetic analog, because that will also give you an idea about the importance of the phytochemicals. Not that every time the phytochemical which is present in the plant is of the important, but their derivatives which have been made from that, those are more active, and many a times actually you may get altogether different activity from them. Well, this is what the driving factor. Actually, I also started extraction and isolation of the phytochemical, and this was the driving factor for me because they are pretty expensive. Now, these are the current prices. Beta-myrin. Now, if you look at this slide, beta-myrin is present actually in a plant called Ficus elastica. I don't think any pharmacy person will look at this plant. Arjunic acid, well, is from the terminally Arjuna. Gymnema genuine is from the Gymnema sylvester, which is a gudma, or since we are in Maharashtra, it is called Bedkita pala in Junnar area or Mirashangi. Shatawarin is from the Shatavari or the asparagus. Vasicin is from the Vasaka. Vithafin A is from the Ashwagandha. These are all Indian plants, and if you have to buy phytochemical, these are the rates actually from the Sigma or the Merck. So it is actually not a good situation. These are the Indian plants, and for which if you have to buy phytochemical or the marker compound from the overseas company for the plant which is indigenous to the India, and buying phytochemical from the overseas company is not a good situation. I think we need to change the situation. Accordingly, I have started making some effort. I have at least made some progress. probably I'll make further progress actually over a period of time. But yes, this is the area where people can think about it in getting into it. Actually, more phytochemical you isolate, it's a lucrative business. 1 gram, 2 gram possession is actually a substantial quantity. All right, I'll just give you a few examples. Diabetic is quite prevalent today, and half of our parliamentarians are also diabetic. So any project in the diabetic area is surely to get it funded actually by them. But today we have the major drug which is available in the market actually to address type 2 type of diabetes is metformin, butformin, and the fenformin. These are some of the APIs which are available in the market. But if you look at their origin lies in a plant. It's a plant from the French, which is the goat's rue or the Gallica officinalis. And this particular plant is considered to be toxic in France because it causes hypoglycemia. And that is what is considered to be the toxic. And when the chemical evaluation started, it was found actually that it contains guanidine derivative, which comprises of the prenyl guanidine, that is galligine, and xanthine A, that is actually a digonidine. So these are the two constituent, galligine and the xanthine A, which were associated with the hypoglycemic action. And when the scientists came to know that this is the constituent actually, then the brain started working, and today we have a synthetic analogs in the form of the bigonide. So metformin is an N-dimethyl bigonide, fenformin is the phenethyl bigonide, and butformin is a butyl bigonide. So these are the constituents which have been developed actually because galligine and the xanthine structures were made available, and scientists could think about it actually. And today we have metformin, butformin, and the fenformin, which is addressing the type 2 diabetes. Another example, quinine, and very good example actually. If you look at the structure of this, quinine is the first structure. If you look at this is a quinine structure, and then we have a synthetic analogs, quinaquine, chloroquine, hydroxychloroquine, mefloquine, primaquine. So these amodiaquine, these are the various derivatives. If you look at the structures very closely, you'll find one important features in the structure, that everybody has maintained the quinolidine ring, because natural quinine comprises of the two important features. One is the quinolidine ring, and then the quinacridine ring, which is responsible for its pesticide or the insecticide or the mosquito repellent action. And today we have actually all the synthetic analogs, and if you look at all the synthetic analogs, they have retained both these features in the some or the other form. Like quinolidine ring is retained in all the form, then only it was found to be associated with the antimalarial action. And quinacridine ring also in some or the other form, it has been retained. And that is why the compound which has been synthetic analogs, they were also found to be associated with the similar kind of the action, because they were heavily influenced by the structure, natural constituent which is found to be present in cinchona. Another example and good example, steroids are very difficult to say. If you have to start steroid building right from the step 1 in the lab, it is very, very difficult. Probably it may take more than 25 steps to synthesize steroid, and then of course the quantity what you will get it actually after 25 steps. Well, I think any organic chemistry will be better know about this. But then we have a natural option available. Dalgenin is perhaps actually the best option which is available. Natural, of course, there were 3 options available earlier: dalgenin, hecogenin, and solasodin. Out of which dalgenin still remains actually very active and major source, because dalgenin is present in a diascorea plant, and it can be conveniently converted into a 16-DPA, that is 16-dehydroquinone acetate. And once it has been converted into a 16-dehydroquinone acetate, which is just this 2-step reaction, thereafter it can be easily converted actually to the different steroids, which includes hydrocortisone, betamethasone, dexamethasone, which are important corticosteroids and extensively used as a anti-inflammatory drug. So again, the origin lies actually in the plant called Diascorea. Another plant material, morphine, and today I'm here in Gujarat. I'm 40 kilometers away from the Baroda in an industrial area called Sauli, and I'm working on the morphine. I will not be able to reveal further about that, but some information I will reveal. Morphine and synthetic morphine, government, this is entirely the entire morphine trade in the country is being controlled by the government of India, and the constitution which are present, the entire thing is being controlled right from the issuing the permit for the cultivation, and then collecting all the opium, Now, here I am associated with developing a method for the extraction of opium alkaloid from the poppy straw. Poppy straw is a fruit uh, which is of the poppy, wherein actually the khaskhas, that is the seed which has been recovered, and then actually this is unlanced. After opium is removed, actually that poppy fruit is useless because after lancing, there's hardly anything which has been left. But what I'm talking about actually poppy straw refers to the fruit actually which is not given the cut but allowed to ripe, and the seed actually which represent khaskhas is useful, whereas the straw contains still 0.6 to 0.7% opium alkaloid. So now I'm developing, and that is a huge tonnage which is available in India. Every year some 10,000 ton is available. So now government has decided to make use of that, and accordingly I am developing actually the process for extraction and isolation of the opium alkaloid from the poppy straw. Well, these are some of the synthetic analogs. Morphine, of course, is a very powerful painkiller, and it is still used in the limited fashion today in the form of the morphine sulfate injection, especially the post-surgery. Heroin is another very simple derivative and which is quite popular with the unethical or the illicit world. But yes, it is a derivative. And then we have a synthetic analogs. Heroin is actually a derivative, whereas the fentanyl and the sufentanyl are synthetic analogs. And if you look at the activity wise, if you take morphine as a one, then heroin is having the potency which is twice as that of morphine, and fentanyl is having the 100 times, and sufentanyl is 500 times. So synthetic analogs are more powerful, but at the same time they are also opioid because they also have the same problem associated with them as that of morphine, and with the result they are both manufactured and the trade is being controlled. Menthol, and this particular slide is specifically targeted for the researchers how to think in the right direction. This was the exercise which was given by a company called Wilkinson Sword in US, and they were used they're into the cosmetic, and they were using coolant menthol actually in their product. So they have put their team actually in the action that in case if the menthol is not available, how we will be able to manage the cooling sensation or the cooling effect. Then the different strategies got evolved, and the first strategy was actually look within family. Within family, that means menthol is a monoterpene. So like that, another monoterpene, you find out whether they're also having cooling sensation associated with them. Then came actually thymol, camphor, and the cineol. Thymol is present in the Thymus vulgaris. Camphor is from the cinnamon camphora, and the cineol is from the eucalyptus. All of them, they do have a cooling sensation associated with them, but less in comparison to the menthol. If you see here the scale, actually, you'll find actually thymol is having 25, if the menthol is 100, thymol is having the 1/4. Cineol is slightly more, but they are having the much more lesser cooling sensation associated with them. But they have a more drawback. They are also having very characteristic smell and the taste associated with them. So that limits their application to substitute menthol. So the second strategy, all right, you have a menthol, but it is less menthol is available. So how to make use of the menthol in very judiciously. So the strategy came actually with the result that let us find out what are the drawbacks of the menthol. And one major drawback of the menthol is actually the action doesn't remain for a long time. So when you pop in any mint or polo, the action remains at the most for the half an hour. Thereafter, the action subsides or it vanishes. So how to improve the action or how to make the action lingering with the same quantity of the menthol, that was the effort has started. And then they started making the molecule bulkier. That means you make molecule bulkier so that it will be less volatile and it will give the cooling sensation for a long time, though it may compromise some cooling strength. And accordingly, some derivatives have been found out. Methyl succinate, paramenthen carboxylic acid is a very important intermediate which has been made from the menthol, because once it has been converted to paramenthen carboxylic acid, it opens up a lot of possibility of converting into a derivatives. And accordingly, WS3 has been developed actually, which is actually ethyl carbamide. And if you see the WS3, the cooling sensation is 150. That means it is having the more cooling sensation than the menthol. At the same time, more lasting sensation than the menthol. So the second strategy, wherein they were able to convert this menthol into a paramenthen carboxylic acid, and thereafter they were able to make it more heavier, it has spread to them, and they were able to come out actually with a molecule which is more coolant and more lasting coolant than the natural menthol. And the third strategy is without menthol. That means now there is no menthol, it is totally a synthetic analog. but certain features like molecular weight, they have confined to the same molecular weight that of menthol, plus the spatial arrangement of the various groups actually, and they were able to come out actually with the amide, which is also having WS23. WS23 is also having the cooling sensation, though not as menthol, but it is totally synthetic. So it is possible to have a synthetic molecule having the cooling sensation without menthol. So these are the strategies on which the company has worked. And today, I must tell you, there are 50 different derivatives of the menthol which are already in the market, which have been used in either food sector or in the pharma sector or in the nutraceutical or even in the cosmetic sector. So this is the development which has led to the vast development of the derivatives and the synthetic analogs which are extensively being used. Cathinone and the synthetic analogs. Another important, narcotic plant. Both cathinone is from the Catha edulis. It's an African plant. The local people, they use it chewing leaf. Actually, they chew leaf like we use betel leaf here. And ephedrine is from the ephedra. I think that does not need any further explanation because it's an undergraduate material. So if you look at the structure, actually they are phenylpropanoid. Amphetamine is a structure actually. And if you look at the other derivatives which have been listed here, methamphetamine, methcathinone, methadone, that is 4-methyl methcathinone, and esketamine, that is MDMA, that is 4-methylene dioxide, methamphetamine, and methadone. These are all party drugs actually, and of which the idea has come actually from these two plants, cathinone from the Catha edulis, that is actually a cat plant from the Africa, or ephedrine from the Ephedra vulgaris, actually a plant which is extensively growing actually in the Indo-Tibetan region. So this is the plant which has laid down the foundation for the development of this party drugs actually, and which are today extensively being used as, abused actually. I'm not saying use, abuse. And there's a control on that. All right, so those are all the derivatives actually which were used for the same purpose for which they were present in the plant. And here is the derivative. This is a barbaloin from aloe, which is having the purgative action, and it has been converted actually. So barbaloin, which is a glycoside, is converted to a glycone by oxidative hydrolysis using ferrichloride and the hydrochloric acid, and it gets converted into aloe-imodine. Aloe-imodine, then it is oxidized to this CH2OH group is oxidized to make it carboxylic acid group. Then the compound is called rhine, and then these two 1,8 hydroxy group are acylated, and then the compound is called 1,8 diacetyl rhine, and that is called diacerine. Now today diacerine is an API which is included in the pharmacopoeia, Indian pharmacopoeia, and diacerine is having altogether different action as that of starting material. Starting material is purgative, whereas diacerine is anti-arthritic drug. So even there is a possibility that the derivative may have altogether different pharmacological action than the parent molecule. All right, this is one more example. All of you are using some of the other mosquito repellent at home, and they all have been influenced by the naturally occurring pyrethrum. And all of them, if you look at the structure which has been used, the two constituents which are being used actually in the household mosquito repellent is transfluethrin or dialethrin. And both of them, if you look at the structure closely, they both retain actually the same feature as that of natural pyrethoids, which is actually a dimethyl cyclopropane ring, as well as ester linkage, which has been pointed out here in the red circle. And if you look at other molecule, they have the same feature actually being maintained. And on either side of this, actually, the same bulkier group is also been maintained. And then only the compound is found to be associated with the mosquito repellent action. All right, now I will just another 10 minutes at max. I will take few case studies which will give an idea. These are all commercially important plant material of which I have undertaken in the laboratory and developed method for some of the other industry. That will give some idea. Ellagic acid, since this particular FDP is in Pune, I have undertaken this project actually for Adivasi cooperative Sangh, Kukareshwar Adivasi Hida Utpadak Sangh, which is situated in the Junnar, and they have a good amount of Hida production in the area because everybody has in their backyard the Hida tree, and they have a good amount of Harda every year being obtained. So they came forward actually and they wanted to add value. Plant is ready actually, and there we have put up the plant actually for both tannin as well as ellagic acid. So ellagic acid is a compound which is not present in a high concentration in the harda as such, but ellagic acid can be conveniently prepared actually from the extract. Ellagic acid is actually a dimer of the gallic acid, and ellagic acid can be prepared because the harda contains, or the terminal chibula contains ellagic tannins. On hydrolysis, it gives ellagic acid. Ellagic tannins can be conveniently hydrolyzed by sulfuric acid, and advantage of the ellagic acid is that it is insoluble in water. The only problem associated with the preparation is that since the fruit are starchy, you need to be very careful. Extraction need to be carried out in the cold. You cannot use the heat, because if you use the heat, the starch which is present in that will get gelatinized, and the subsequent operation of the filtration will be a very messy, or probably it may not happen. So with the result, you have to do the extraction at the cold, so that the starch which is present does not get gelatinized, and you get the extract. Though extraction is compromised, but you get the clear extract, and then of course you have to undertake the hydrolysis for a couple of hours using sulfuric acid. On on hydrolysis, you'll get the ellagic acid, and the advantage is that ellagic acid gets freed from that in the form of the precipitate, which can be further purified. And the gallic acid is also formed that image into the solution, and that can also be separated actually by ethyl acetate extraction. So from this, you can get two important products: gallic acid, which is extensively been used in the food industry for the making gallet, and ellagic acid, which goes into the nutraceutical field. Henna, this project was undertaken by Godrej. Henna, all of you know, Lawsonia inermis, Literaceae family, and Godrej is having the hair dye based upon the henna. They are using the powder as such of the henna, but the company wanted whether we can make concentrated form, so that people need to use less quantity and they will have more room for adding other constituents. So they approached me actually for 20% Lawson content in that. And we were able to develop a method actually. Again, the problem what we faced was the filtration problem. What we did actually, we treated with alkali in cold, and anything when you use water as a solvent, you need to use substantial times, almost 10 to 12 times as that of the starting material, and which causes a lot of burden of the filtration. In this particular case, henna is also having a lot of problem associated with that. It contains mucilage, so filtration was a major problem, and we were able to overcome that by adding some chelating agent. But yes, filtration remains the problem. And afterwards, once it is, and another problem associated with the henna is that the Lawson is not present in the free form, but it is present in the glycoside form. So you need to convert that glycoside into a glycon. That means it should get converted into a free glycon. Then only it will be soluble into the organic solvent, and you will be able to remove that. So by doing this, we were able to undertake actually the extraction and the enrichment to the 20%. And today we have also developed a method to go right up to 85 to 95%. Another study, kaolin. Kaolin is from a plant called Ammi Visnaga, Umbelliferae family. Now it is of course renamed as IPSC family. And this of course I have undertaken actually because I was working on the monograph preparation and we wanted to isolate kaolin. So we extracted the isolated kaolin with the help of, it's a furanochromone derivative. And yeah, you can see the furanochromone derivative it is. And quite simple extraction process. The extraction in the methanol, then concentration, concentrated extract, then you fraction it with the petroleum in which it is insoluble. And then of course it gets separated in the crude form, which need to be further purified by the repetitive crystallization, and then you get the purified kaolin. We were able to publish a method. We were able to get the good purity, publish a paper, and fortunately our paper got accepted actually. Our paper has been listed actually as a reference in a wiki. Another case study, very common plant, Mangifera indica. And again here actually, unfortunately, we are importing Mangiferin, though in the entire country we have a mango trees, some or the other variety growing in entire country. But still we are importing Mangiferin from China, which is not a good situation. Mangiferin is present in the bark. More precisely, those who are from the botany or from cognosy, it is the inner bark which contains more of Mangiferin, which is actually a xanthone derivative. And again, simple method of the extraction, not very complicated. It is insoluble in water, so extract with the methanol, and then of course concentrate in the precipitate by adding into the water. It gets precipitated, but you also get good amount of the lipid or the fatty material being extracted from that. So you have to wash it actually with the pet ether, and then of course, though of course it is represented in very simple fashion, 4 steps, but actual reality actually the more steps, because as you go on enriching the purification or the purity of it, you need to actually give a number of steps more. But yes, this is actually a schematic diagram wherein actually these are the steps involved. Of course, you need to do repetitive steps many times. Just the confirmation. So I will not go deep into that. Another project, we were able to, in situ, we were able to generate aloe without separating barbalone from that. So what we have aloe a juice, or the dried aloe juice, that is actually aloes of the pharmacy, which contains around 20-22% of the barbalone. So instead of separating barbalone, we developed the method actually that in situ we undertook actually oxidative hydrolysis, and we were able to separate aloe from that. And we were also able to convert aloe into a Rhine, but further step we did not do it actually because industry was not having actually the license for the acetic anhydride, which is controlled item by the again because of the narcotic. But we were able to convert actually to aloe and then of course to the Rhine part of it. Another case study, ursolic acid. Ursolic acid is present in many plants like vinca, tulsi, but we came across a toxic plant called Nerium oleander, which is toxic plant. But the toxicity is more in the root part of it, which contains actually oleandrin. Oleandrin is actually a cardenolide, that is a C23 type of glycoside, which acts on the heart, and that is the constituent which is responsible for its toxic action. But what we did actually, we made use of the leaf, which contains high concentration of the ursolic acid. Ursolic acid incidentally is also present on the apple skin, and to great extent actually it acts as a protective. And we were able to develop a method actually for the extraction isolation of the ursolic acid from leaf in a high concentration, almost more than 2% concentration, though in leaf it is present almost to the extent of 4%. And we made use of the carboxylic acid group for its purification. That means you convert into a soluble salt, and once again by adding equivalent amount of the acid, precipitate, and that is how, since it is obtained from the leaf, chlorophyll becomes a major pigment which interferes actually in its purity part of it. So we have to repeatedly undertake this dissolving into the alkali and then reprecipitating with the acid. This is just the confirmation of it. I think this is another case study for scoline, wherein we use initial part of it supercritical extraction, because this root contains around 0.5% forskolin, which is an anti-obesity drug. And but the root also contains volatile oil and the resinous material. So if you extract with the straight away with the methanol, you get a lot of impurities. So what we did actually, we first initially extracted by the supercritical at the low pressure, that means 100 bar pressure, and without any entrainer. So it has favored more non-polar, that is only lipophilic part of it. That means volatile oil as well as the resinous part got removed. Subsequently, actually the pressure was increased to 350 bar pressure, and also the entrainer was added in the form of the 1 molar percent of the alcohol, ethanol. And then it favored actually the extraction of more forskolin. Then that extract was concentrated, and then of course it was repeatedly dissolved into the chloroform and precipitated by adding pet ether in that. And number of times you perform that, then you get actually a refined forskolin, and we were able to reach purity of 98% from that. This is just the confirmation of that. Karanjin I will skip because there's no much of time. Karanjin has a case study. This is the case study of the 20-hydroxydiazone. That is what the project I said from the Kaladana. This is an important hormone in the life cycle of silk insect. These are the 5 instars. So from the larva to the pupal stage, see when it starts spinning the cocoon, actually it has to develop. So from egg to larva, and the larval stage has to grow. So it has the 5 instars in which it grows. So each instar lasts for the 5 to 8 days actually, and this instar where it grows in the size is been controlled by the ecdysone hormone. And in natural course actually this has been derived from the mulberry leaf. However, that is not consistent, and with the result actually the maturation of this is inconsistent, and the small farmer especially faces a problem. So in order to have the consistent development, you need to spike this mulberry leaf with the additional amount of the ecdysone, and that is what precisely the company came to us, and we were able to isolate or enrich ecdysone actually from Kaladana, which is an important hormone in the development of a larva of a silk insect, because only the fully developed larva starts spinning the cocoon around it. Okay, so this is from the Ipomia Hydericia cannabina seed we were able to enrich, and this is just the academic work more. But now here actually the industrial work where we were able to lay down actually the procedure for the large scale. Then we undertook the field trial. These are the field trial we have undertaken actually on 1 lakh insect, and eventually we were able to come out with the product called Sampurna, which is a vial, sorry, an ampoule, and this 5 ml ampoule is enough actually to dissolve into the 20-liter water, and that is enough actually for the 500 kg mulberry leaf on which the insect feeds, and that ensures uniform development of the insect. So Martin, case study, I will straight away go to the final slide because that is important slide. This is just the scheme. Major challenges because this is a protein. See, organic compound is that way very easy to separate, but the protein is very difficult to separate from the plant because they are water soluble. You cannot use any solvent. You cannot use any heat because they are heat sensitive. Beyond 65 degrees, you cannot heat it. You cannot use any chemical actually by which they get reacted actually. So no chemical. So eventually we are left with the only choice of water. And when you use water industrially, normally for the extraction, water is less preferred because it extracts a lot of junk. And later on, since latent heat of vaporization of the water is very high, the removal of the water is very taxing to the company. So company normally prefer not water, but any organic solvent, especially ethanol or IP or acetone. Those are the solvents which are normally been preferred actually by the industry. But here in this case, this was not possible. So we have to use for 5 kg, we were using 1,200 liters of the water. Of course, the constituent is quite expensive. And the molecule is of the 21 kilodalton kd. And we have to use 2 types of the filtration. Again, in this case, we have to first filter actually by first filter the crude filtration and thereafter undertake the membrane filtration. And finally, we use actually 5 kd membrane for its filtration because our compound was 21 kd, so it was detented actually, whereas all other molecules which were smaller than that, they were able to pass through that. And we were able to separate, and thereafter it was subjected to the freeze drying, and we were able to overcome. One problem was the mucilage. We were able to chelate that mucilage with the inorganic molecule, and second one was protein. So we did not use heat, but we used large volume of the water to extract that buffered volume. All right, so those are some of the case studies which I have encountered. And in a process, I have realized actually when you want to undertake phytochemical, there are many challenges, no doubt. First of all, you need to have a mindset to get into this particular field. Facilities, yes, you need some basic facilities, but you don't need very exorbitant facilities. Even today, I manage most of my work with a TLC, and only at the latter stage, when I reach to the final, that time I make use of the HPLC to just confirm whether it has come as per my expectation or not. Extraction processes, I still work with most of the traditional or conventional extraction processes rather than modern extraction processes. You may call it like supercritical or accelerated solvent extraction. I don't use them. I mean, unless it is compulsion. Identification, again, as I told you, you need to identify a right plant interior in the right season so that whatever effort you make it actually, it must get rewarded. It must have a high concentration. Purification is that way, unlike organic molecule purification step, here is many steps actually. It's not a single step purification, but you need to do the multiple times. Time consuming, yes, it is frustrating. Time consuming ultimately to the frustration. But if you remain determined, actually, at the end, it is rewarding as well. So you need to have some patience actually to undertake this kind of work. So never give it up. Good things always takes time, and I have experienced this actually. I remember my first student undertaking the column for 6 months. But at the end of the 6 months, he was able to isolate 6 compounds, and one of the compound we were able to commercialize that as well. So yes, it is frustrating, but at the end, actually, you get rewarded as well. That's all about, these are the phytochemicals which we have isolated in our lab. And we also came out actually one small booklet kind of thing for the undergraduate to practice in order to make it more popular, case studies by which you can practice in the lab and make it, remove that phobia from the student as well as from the faculty that it is not possible. So most of the cases we are used just 100 gram of the starting material, and we were able to extract 1 gram of the semi-refined product. These are some of the industrial projects which we were able to take it into industry, from the lab to the industry. These are some of the, we also associated with the monograph making. These are some 60 monographs have been published by the ICMR in the 7th volume. These are some of the products which we have been able to make it in our laboratory for the different clients. And that's it from my side. Effective development of the technology for extraction and isolation of the phytochemicals will lead their application not just in pharmacy, but in other industry as well. So one should keep their option open. Don't necessarily look only for the pharmaceutical application, but whatever chemical, one thing I can assure you, actually, whatever chemical you isolate from the plant, it is very complicated. And I must tell you, it must find some application. It may not appeal to one sector, but it will definitely appeal to the other sector. So you need to find out that right sector to whom it will be appealing. That's it. Thank you very much. And I sincerely thanks all the participants, those who were patiently listening to me, staff of the AISS College of Pharmacy, faculty, principal sir, and of course, Damle madam, I think whom I was able to connect. Thank you once again to all of you. And now I'm open for question in case if they are.
Thank you, sir.
Thank you, ma'am.
It was really interesting to hear about the experiences and a large scale work that you have done in this industry. Definitely, we have questions for you. Damle madam has a question that is, what should be the choice of markers for quantitative analysis? Active constituent or major constituent?
On the lighter side, shall I tell you? On the lighter side?
Yes, sure, sir. Yeah, whatever is affordable.
No, not affordable. Whatever will give you the money.
Okay.
On the lighter side, yes. On the lighter side, yes. I mean, I must tell you, any compound you isolate from the plant, which is not very obvious and regular, definitely it will appeal to someone in the sector. Okay, and it will fetch price because all the phytochemical which you isolate from the plant are pretty expensive. Yes, you need to have a good purity of that, and then definitely people will make use of that because see, the researchers are constantly looking for newer and newer molecules which are complicated. Okay, and plant provides that option, that advantage, And well, when you look from the pharmaceutical angle, yes, active marker is the right one. Actually, it must be associated with some biological action. Those are the marker compound which should be chosen actually when we talk about pharmaceutical or pharmacological action wise. So that is a marker compound. We should make use of that. But in case if it is not, then you should see the another requirement of the marker is actually it should be unique also. It should be unique to that particular plant. In case if the one compound is not unique, then you should take two compounds from that plant, because then the possibility gets reduced actually. In case if the two is not unique, then you must take one more three. That is what precisely Chinese pharmacopoeia offer. In some cases, they have mentioned one marker, in some cases two marker, in some cases three marker. Two only by qualitative and one by quantitative. Any other question?
Yes, sir. We have one more question. I would like you to highlight on the challenges in achieving the stability of color pigments. Your initial slides you had shown about the coloring pigments that you had that are used in food industry. So
Yes.
the stability of these coloring pigments is definitely a challenge. So how do we deal with it?
It is a challenge. We have two types of the colorant. One is water soluble, and the other one is actually the oil soluble, or the lipophilic and the hydrophilic. As far as lipophilic is concerned, actually, all of them are very sensitive carotenoids. Majority of them are carotenoids actually, and carotenoids are very sensitive. But yes, at the same time, you must find the right application. For example, as I told you, carotene is used in the Amul butter, which is to be refrigerated. So their stability is not a problem, and it is extensively used as well. Ice cream is another sector where carotenoids can also be used actually, because once again, ice cream has a lipophilic in that constituent. So I think instead of complaining about its stability, I think one should find the right application that will be more appropriate. Okay. Hydrophilic, yes, there is a problem actually. Whether you take flavonoids or whether you take any phenolic compound, they have a problem because they are not stable. Any phenolic compound, when you take it, they have a problem of oxidation over a long period, and you are not making a product actually just having small shelf life. So definitely on aging, they have a problem actually. To some extent, they can be stabilized by concealing. Make it actually a microencapsulation. That is one way of, if you want to increase the stability, microencapsulation is one thing actually. Or the OmniActive has come out actually with a beadlet. They have a carotenoid beadlet. So their stability is enhanced definitely. Not, it is, I must say you, it is not completely made stable, but definitely that process of instability has been lengthened. So now what it was having the lesser shelf life, now we have a better shelf life. But yes, you have to deal with this problem actually with the natural pigment.
Okay. Thank you, sir. There's one more question.
Yeah.
Which properties of phytomolecules must be considered for fast and easy isolation of these molecules?
There is no shortcut. Each compound is different. Even if you take it from the same class, and the constituent from the plant in which it is present is having the different composition. Even if you have the same compound in two different plants, they have a two different composition. And what you want is only 1% or less than 1%, but what you don't want is 98 to 99%. So many times you have to look at the what you don't want it rather than what you want it. So other things which are present in the plant also need to be looked into it, and sometimes you have to address your extraction process in which they are soluble, remove them first, and then you look at the one which is not, which is of the interest. So these are the something actually, and see, there is nothing like universal extraction process because each candidate and from each plant it is a unique case. So depending upon the composition, depending upon the concentration at which it is present, you need to develop a specific process for that.
Okay, and this is the last question. Marker compounds or the phytoconstituents, isolated phytoconstituents price is a major issue in standardization. Say if at all a student goes for formulation, in order to evaluate that formulation, the marker compound price is always an issue.
Yes, it is. And it's going to be issue till people don't work in that direction.
Okay.
That is why more researchers should work into that and develop technology. Yes, we have a constraint that it does not present more than 0.5 to 1%. But today, those are the, I'm extracting actually 0.6% opium alkaloid in a commercially feasible method. So it is not that it is not possible. Only need the, as I told you, yes, it is the process actually which is not, see, we've seen the problem with the student actually, they want degree within a month. Okay, that is not possible. You need to actually work because you need to understand this first, which takes time. For the last 3 months I'm working, and even today I'm not stabilized, on this. But yes, every passing day we face a major, newer problem. So we need to address them actually, and you must have that patience and the stamina to address. And at the end, today we have isolated actually a morphine, and we have given to the government lab, and they also started wondering actually we were not able to successfully do that. How you are able to do this actually? So now we are trying to improve our method actually because they are very happy with that. So this is what will happen. This is what will happen. You need to involve, and even teachers need to be involved actually in this. Unfortunately, what is happening, I'm sorry to say, but majority of the teachers actually they remain confined to the cabin. They must enter into the lab. They must see what students are performing. If not, they should do it actually. I think the teacher should also be working in the lab. I have put this criteria for me, and even today I'm 63, but every day 4 hours I work in the lab.
That's awesome.
And I think sometimes you should reduce your administrative work and get into the lab because that is more rewarding. And I think this problem of making available phytomarkers to some extent it will be I think addressed. And at least for the Indian plant, we must work.
Yes, definitely.
All right. Thank you very much.
Thank you, sir. Thank you for your valuable time.
I have to go actually for monitoring certain parameters.
Sure, sir. We will conclude with this. We are thankful on behalf of management of AISSMS Principal Dr. Ashwini Madgulkar and IQSC Head Dr. Mrinalini Damle. Thank you for being a part of this session. Thank you very much.
Thank you very much, madam. Thank you. Namaskar to all.
For the participants, I would like to announce that there would be a lunch break till 2:30, and we shall resume with the next session at 2:30. Thank you.
Participants will join with the same link at around 2:25. Thank you.